Welcome back to the cell signaling course. In last class we were discussing how different types of receptor tyrosine kinase signaling initiates at the cell surface and meets its goal. One of them was JAK-STAT pathway, and if you recollect, I paralleled JAK-STAT pathway to more like a two-component system which we have seen in microbial world as well as in plant kingdom. For the same reason that there are two distinct components, one which is receptor binding kinase and the other one is kind of response regulator which we saw as a STAT molecule. Today we will see more details of JAK-STAT pathway, where it works, how it is regulated and what goes on by the effector molecule which regulate the JAK-STAT signaling. Before that, let me remind you that how downstream of the activated receptor, which was receptor tyrosine kinase, different molecules come into picture. One set of adapters, one set of scaffolds, and there are molecules which are called transcription factors. Remember, for all of them there is one domain which is mandatory, which you see here. This domain, if I label it as SH2 domain, you can also say in addition to SH2 domain, it could be phosphotyrosine binding domain. Why? Because SH2 is one of the first domains identified which has the affinity for phosphotyrosine. Since they are working downstream of the receptor tyrosine kinase which is activated, and one of the first thing upon activation that happens to the receptor is autophosphorylation, generating phosphotyrosine. So any signaling to move forward must utilize this domain containing proteins to take signaling forward. So there are three types of molecules or effector proteins which will be coming into picture. One will be adapter, and we saw example of GRB2 which had SH2 domain by default, and there were two copies of SH3 domain. And these SH3 domain containing proteins, they interact with polyproline sequence which may come in this motif. And we saw example of this, through which the RAS-GEF was activating RAS and activating the MAP kinase pathway. The other set of molecule with SH2 domain which attach or bind to the activated receptors are scaffold class of molecule which bring additional domain like pH domain. If you remember, these pH domain have affinity for phospholipid and more specifically PIP3 kind of lipid, where you have phosphoinositide 3,4,5 trisphosphate. So in the membrane, wherever you have this PIP3 kind of lipid, pH domain will help these proteins anchor to the membrane region, and the SH2 domain which will be there will help recognize the phosphotyrosine on the receptor. Then you will have transcription factor like molecule, and this is where we discuss the STAT like molecule which is the component of JAK-STAT pathway. So in addition to having the SH2 domain, you have a DNA binding domain and transactivation domain, which is written here as TA domain. So basically this kind of molecule will serve as a transcription factor, and a STAT is also called signal transducer and activator of transcription. We will see more of this particularly with respect to JAK-STAT pathway in coming slides. So if you recollect, the JAK-STAT pathway which I mentioned briefly in the previous class was that here the JAKs are kind of free kinases, and these are tyrosine kinases. So in this case the receptor does not have a intrinsic kinase activity, rather it recruits an enzyme which is here called Janus associated kinase or JAK. So once you have the ligand available, the binding between JAK and the receptor is strengthened, and you end up seeing the cross autophosphorylation of JAK, which phosphorylates the receptor. So, these are phosphate moieties which are attached over here. This phosphate moiety will be recognized by STAT molecule through its SH2 domain, and this brings SH2 domain containing STAT molecule in proximity of the JAK kinase, which now ends up phosphorylating the STAT like molecule. And this phosphorylation on STAT decreases affinity of SH2 domain with the phosphotyrosine on the receptor. What happens? There is a reorganization and the STAT forms a dimer, and this dimeric STAT goes inside the nucleus. So, you can obviously say this must have some sort of nuclear localization signal, and here it activates the transcription of gene expression. Here it activates the gene transcription, which will be mediating in many cases cytokine response. Now, if you look at the components of the JAK-STAT pathway, there are two main molecules which are JAK-STAT, and then there are regulatory molecules, because you realize that JAK once activated can phosphorylate the receptor and STAT can be phosphorylated further, and the signaling will continue. You need certain regulation on the signaling, that is where they are additional components, and these domains and these proteins have very modular arrangement of domains like JAK. So, it is associating with the receptor, so it has a receptor interaction domain. If I can say this is a simple arrangement of domains from N terminus to C terminus, then you have a receptor interaction domain. Obviously, you have a kinase domain, which is again replicated, but there is a change or mutation because of which it is called pseudokinase domain, which is present in addition to the kinase domain. And with respect to the JAK-like molecule, they are also referred as JAK homology 1 domain and JAK homology 2 domain. We will see the significance of these domains when we discuss more that how JAK-STAT signaling is regulated. Then we have a STAT molecule which has the oligomerization domain. Obviously, you saw the minimum dimer-like STAT formation was happening, which was making it move into the nucleus. So, it should have a protein-protein interaction dimerization domain, and to mention here that all these molecules, they recognize and fall in place by virtue of having this SH2 domain, which will recognize the phosphotyrosine residue. So, a STAT also has this SH2 domain and DNA binding in transactivation domain. So, basically it will have dimerization, oligomerization domain, then you have DNA binding domain and a transactivation domain, if I can say TAD. Together these make the transcription factor minimal region. So, JAK will be serving to bind to receptor through the SH2 domain which it has, and then it will be activating the STAT molecule. This is how signaling will continue, as we saw in the previous slide. STAT will move into the nucleus and reply to the transcription processes which are required. Then there are regulatory molecule, one which is called suppressor of cytokine signaling. Here you have a kinase inhibitory domain, which basically means that it will affect the JAK activity. So, if I say Ki domain, so basically it will affect the kinase activity. Then in addition it has the SOCS domain, which is also called that it has a E3 ubiquitin ligase binding domain, and this domain will allow the ubiquitination of the molecule to which it is binding. So it is binding, first of all, it has the SH2 domain also. So it can bind to the receptor and ensure that the kinase activity is inhibited, and if required, the other molecules or the JAK or the receptor itself undergoes degradation by ubiquitination process. Then there is another molecule, the fourth molecule in this pathway, which is called SHP domain, which is called SHP protein. It has two SH2 domain. Basically, it can engage more vigorously with the phosphotyrosine which is generated either on the JAK or on the receptor itself. And then it has protein tyrosine phosphatase domain. Basically, it will recognize the activated receptor or activated kinase and perform the phosphatase activity. What this phosphatase activity does, it basically will remove the phosphate group. So basically, if you consider that it has two SH2 domain, and then if I say protein tyrosine phosphatase, if this domain is present, it will recognize the phosphotyrosine residue and generate the free tyrosine, and a PI will be released. So together they will be working in the JAK-STAT signaling pathway. So simply you can imagine that JAK-STAT positive signaling. If you talk about SHP and SOCS, it basically takes the signaling in opposite direction, no signaling, or it is kind of interregulation where the signaling is regulated. Now this is what I showed you in the previous slide, that you can have, apart from its targeting into the nucleus and catalyzing the transcription process, it will be SOCS which will inhibit the receptor phosphorylation and the ubiquitin mediated degradation of the molecule, and SHP, it will dephosphorylate the JAK molecule and downregulate the signaling. So you understand that how nice feedback mechanism is playing. It is also possible that SOCS is being transcribed in response to JAK-STAT signaling. So this will serve as a negative feedback. So JAK-STAT pathway will be activated in multiple different ways, and it will be ensuring that the signaling is moving forward, and when required, it is sent in the backward direction through feedback regulation. So I was telling you about that JAK-STAT could be regulated by the presence of one of these domains which we call JH or JAK homology domain. You recollect that SOCS and SHP are doing the regulatory function over there. There could be regulation on the STAT molecule. We know that its phosphorylation is critical, dimerization is critical. But what is happening at the JAK level itself? JAK has a domain arrangement where you have multiple domains. We saw earlier that it has a receptor binding domain, it has a SH2 domain, it has kinase domain, and it has a pseudokinase domain. So here, consider this as the C-terminal tail of the JAK molecule. The two domains are here called JH1 and JH2. So normally, if this is the arrangement of JH1 and JH2 in the C-terminal tail, when they are not active, they are kept in inactive state, which means that there could be an orientation where the JH1 domain folds back onto the JH2 domain of the same JAK molecule, the scenario which is shown here. And remember, JH2 was more like a pseudokinase domain. So the interaction between kinase and pseudokinase domain keeps the JAK molecule in inactive state. That is why it is inactivated. And this will be preferred when signal is absent. Other way of inactivating this JAK molecule is where the two JAK molecules which are in the vicinity, they can actually engage in this cross inhibition where the JH1 of one of the JAK is inhibited by JH2 of other JAK. Similarly, JH2 of the first JAK molecule is inhibiting the JH1 of the other molecule. So, it was self blocking of the activity or inhibiting the activity of the same JAK molecule, and this is between the two molecules. So, this is intramolecular and this is intermolecular inhibition of the molecule. Now, when ligand binds to the receptor and there are events which happen with relation to conformation change, the phosphorylation, the JAK is activated and the phosphorylation happens on these domains. So, phosphorylation will be initiated because of these conformational change. So, what conformational change you are expecting here? That this kind of intramolecular or this kind of intermolecular inhibition which was there, that will be relieved. So, basically what will be happening? You will get arrangement from either this or this scenario to an activated state where JH1s will come and pair. So, JH1 rearrangement will activate JAK. Now, this JAK, as we have seen, will be able to phosphorylate the receptor. That receptor will bring the STAT molecule. STAT will also be phosphorylated. It will dimerize and then it will go inside the nucleus and carry out the transcription of desired gene to meet the cytokine response often, but not necessarily every time, because there are several ligands which are known for the JAK-STAT pathway. So, if we have a look at some of the known ligands and the signaling which is mediated by JAK and STAT combination. So, gamma interferon. Here what is understood through multiple research work that JAK1 and JAK2, they activate STAT1, and what this signaling does, that it activates macrophages and increases MHC protein activation, basically helping in the process of immunity. We know that macrophages, when activated, they can actually do phagocytosis and clear the pathogen. Then there are alpha interferon as a ligand. They use another kinase called TYK2 and JAK2, they work in combination. Here you see activation of STAT1 and STAT2 molecule. So, there is a possibility of STAT1 homodimer and STAT2 homodimer as well as STAT1 STAT2 heterodimer, which can actually activate the signaling, and this will induce the resistance to viral infection. So, basically antiviral molecules will be produced so that the cell is gaining resistance against the viral infection. Then there is a molecule called erythropoietin, which is important for the blood cell formation, where JAK2 is involved and the STAT5 is involved. What it does, it actually stimulates the production of erythrocyte. This is what I was mentioning in the previous slide, that not necessarily every time cytokine response is mounted. It is dependent on the ligand which was bound, like you saw in case of interferons. You went into a immune pathway activation where cytokines play a very important role. Then it is also known to bind to the prolactin molecule. This is the hormone which actually activates the milk production in mothers. So, here JAK1 and JAK2 again come, but you see now it is in combination with STAT5 and it stimulates the milk production. Remember, one of the things which I told, that these molecules could be common in the cell, but there will be specific molecule which will be destined to do a particular job, like here milk production. Even if JAK1 and JAK2 come in combination with STAT2, it will not be able to do the activation of milk production because the target cell type is not right type. So, target cell dependent response is something which is very important, because same ligand will generate different responses, as we had seen long time back in this course, that acetylcholine was eliciting very different responses in different cell type. Then there is a growth hormone which engages JAK2 as well as STAT1 and STAT5, and it stimulates the growth by IGF production. Remember, this is IGF is called insulin-like growth factor. This works through IGF receptor, it also works through insulin receptor, and together it will stimulate the growth. And if you recollect, it is actually working through receptor tyrosine kinase. JAK-STAT itself is a receptor tyrosine kinase kind of signaling, and we saw how collectively different receptor tyrosine kinase signaling helps in cell growth, proliferation, survival, migration, and so on. Then there are signals which are called GM, granulocyte and macrophage colony stimulating factor. Here JAK2, STAT5, they stimulate the formation of granulocytes and macrophages. Granulocyte, it could be neutrophils, it could be eosinophil and basophil, and macrophages, as we say, these are the most important type of phagocytic cells which are present over there. Then there is something called IL-3, interleukin. They work through JAK2 and STAT5 and stimulate early blood cell production, some set of progenitor for blood cells, different types of blood cells. What you would notice that in these two cases, see, the combination of JAK and STAT is same, however the ligand is different, right? So the receptor to which this ligand is binding versus IL-3, the receptor to which IL-3 will be binding, both of them recruiting the same set of JAK and STAT. However, the responses which are met is very different, because this kind of blueprint is present in the granulocyte and macrophages, and this kind of blueprint is present in a different set of blood cells. So what we can say that we have understood in this class is that receptor tyrosine kinases are diverted to different routes of signaling by binding of the proteins which are in the class of adapters, scaffold, enzymes, and transcription factors. Then we have seen that how JAK, which is a very different type of signaling of receptor tyrosine kinases, because here receptor does not have the kinase activity, rather a membrane bound kinase has the tyrosine kinase activity. It works through JAK-STAT pathway, and it also has a pseudokinase domain which exerts a control on the activation of JAK itself, so that only when ligand is present, receptor is engaged, these JAK kinases are activated. It is also regulated by the phosphatase and the ubiquitination activity, the JAK-STAT pathway, because we saw how one of the important molecule had the phosphatase activity and the other molecule had this ability to induce ubiquitination by interacting with E3 ligases. All the components of the JAK-STAT pathway, they contain the phosphotyrosine binding SS2 domain, which is helping them become congregated at the surface where receptor is getting activated by phosphorylation by JAK. Then there are many cytokines and growth factors which actually engage in JAK-STAT pathway and help in the mounting of cytokine response or growth of a cell type or differentiation of a cell type, as well as activation of cells which are of granulocyte and macrophages. Thank you.